Tasneem madam, please begin.
Okay, sir.
A very blissful afternoon to one and all present here. I am Ms. Rashmi Patel, working as an assistant professor, School of Pharmacy, Parul University. Welcome you all in the national level webinar on incorporation of dissolution data in PBPK modeling for pharmaceutical development, organized by School of Pharmacy, Parul University. I feel privileged to extend my greetings to respected Dr. M.N. Patel, Provost, Parul University, Dr. Abhay Dharamsi, Dean, Faculty of Pharmacy, Dr. G.S. Chakraborty, Principal, Parul Institute of Pharmacy and Research, Dr. Lalit Lata Jha, Principal, School of Pharmacy, and Dr. Aarti Thakkar, Associate Professor, Amity University, Noida. On behalf of School of Pharmacy, Parul University, I welcome you all the distinguished dignitary to grace the online national level webinar on incorporation of dissolution data in PBPK modeling for pharmaceutical development. To enlighten us on the topic, we have Dr. Aarti Thakkar with us. So, Dr. Aarti R. Thakkar is alumnus of Pharmacy Department, the MS University of Baroda, India. She was a visiting scholar at King's College of London, UK during PhD and postdoctoral fellow of UNC Eastman School of Pharmacy, University of North Carolina at Chapel Hill, USA. Doctor Thakkar has more than 17 years of national and international experience in the pharmaceutical science. She started her career as a QC chemist at Illuminous Pharmaceutical Limited, Baroda. She has worked as a faculty in few academic organizations: Parul University, Baroda, ISF College of Pharmacy, Moga, Body University and Delhi Pharmaceutical Science and Research University, New Delhi. At present, she is Associate Professor, Amity University, Noida. In the past, she has developed and established a government CDSCO and NABL approved commercialized drug testing laboratory named as ISF Analytical Laboratory, Moga. Dr. Thakkar has 53 publications in the peer-reviewed journal, 73 presentations in international and national conferences, 5 Indian patent and international book chapter, 6 book for B Pharmacy students and D Pharmacy students, 5 research projects, I AICTE India, INMS India, DST India, NIH USA and J&J USA. Worth rupees 1 crore from Indian and international funding agencies and 11 national and international awards, including IPGA Fellowship Award 2019, Best Research Guide 2012, Award from Progya Pharmaceuticals and Ellen College of Pharmacy, Ahmedabad, and Fast Track Young Scientist Award from DST New Delhi, two-time Best Poster Presentation Award DMPK Symposium, Naipur Mohali, and Best Research Paper Award from PD Sethi Memorial Award to her credit. She is the first certified trainer of the Quality Assurance Chemist job role by LSSSDC New Delhi. She has guided more than 38 in-pharmacy students, pharma analysis, QA, DRA, and pharmaceutics, and currently guiding 5 PhD students. Her core research area are formulation development, DMPK, method development and validation. Current drug regulatory affairs. She is editorial member of the IPGA today. She is life member of IPGA, IHPA, IPTI, SPER, MSU Pharmacy Alumni Association. She is member of AAPS and ISSX. She is Executive Council member in Society for Study of Xenobiotics, India, IPJ and MSU Pharmacy Alumni Association. She is co-convener IPGA Women Forum and Secretary, Delhi Pharmaceutical Council, Women's Indian Chamber of Commerce and Industry since 2020. Dr. Thakkar has organized national and international conferences IPG in 2009, IHPA 2010, IPTI 2011, and DRDO sponsored Indo-Taiwan International Workshop in 2011. DST sponsored INSPIRE Camp in 2012, National Seminar of CRS Indian Chapter in 2016, 17th IPC in 2018, third and fourth annual conferences of SSX India in 2018 and 2019, as LOC member of Scientific Service Committee member, 71st IPC in 2019. She is Scientific Committee member of DISO Research Presentation India in 2020 and 21, is Social for Pharmaceutical Dissolution Science. Today Madam has taken the task of briefing us with her knowledge on incorporation of dissolution data in PBPK modeling for pharmaceutical development. I, Rashi Patel, warmly welcome you, ma'am, in the webinar.
Thank you. Thank you for. Am I audible enough?
Yes, ma'am.
Okay. Thank you so very much for elaborately, speaking up the introduction. Thank you so much. And I'm first of all, I'm very thankful to the management of Parul University, and of course Lalit Lata ma'am. It feels really back home, coming back home kind of a feeling, because I after my masters, I did 1 year job in Parul University, and of course at that time it was a Baroda College of Pharmacy, and then of course, few of my friends or some seniors who are working there, so it really feels back home kind of a feeling. So thank you so much for having me here, and giving this opportunity to talk about this particular topic. So if I say in terms of this topic, what I'm going to talk more or less, we have Rajshri, we have students MPharm and PhD, right? They are the major
Yes.
audience. Okay.
Yes.
So basically, during your maybe B Farm in Biopharm, or if you are from pharmaceutics, you might have heard modeling and simulation, or biopharmaceutics aspect things when you have seen. So this is only that particular topic I'm going to elaborate on. You know, Almost, 2 years, since 2 years, all of us have gone through a very unfortunate time of COVID, and it's unfortunate, of course, we all have witnessed that, but interestingly, that particular time I have utilized in developing some new skill set in me. And when I came back from US, it was more or less work related to drug metabolism, pharmacokinetics, my high-end research, what I have learned in US and during my stay in King's College of London also. But it was very challenging to have that kind of a research, in terms of, not research was possible definitely, but the kind of equipments and all, we all understand that in academia setup, it's bit of challenging happens if you are thinking of having cell culture work, or radio label compound or so. And even if, many times we struggle to do pharmacokinetic study also in animals, the approval process and all. So keeping that all in mind, I thought it would be good idea to go ahead with something different. And fortunately, during this COVID time, as we all were working from home, I got a good chance to am I audible?
Yes, ma'am.
Okay. So, I started working on this software learning skills through some of the people, and I would definitely acknowledge them. Dr. Vijay Vaturi from University of Maryland, Dr. Surli Velrajan from University of Manipal, and, a couple of other people who were part of the team, and I learned these things from them. And then we explored one of the project. So, in this area, specifically developing a physiological based pharmacokinetics model. So let me just share the screen and walk you through some of the work what we have done, and some of the case studies I have taken over here. Okay, so is the screen visible?
Yes, ma'am.
Okay, all right. So So, the title of the talk would be, of course, looking upon the dissolution data in PBPK modeling for pharmaceutical development. And, in terms of Rajshri, I have one question because, I'm able to see just my screen. I'm not able to see anything on screen other than that. So I hope if I get disconnected in case, just let me know through
Sure.
message or something so I can, rejoin also. Yeah.
Okay.
All right. Okay. So things are like, if you just, we all know that, in terms of India, we work a lot in terms of research, pharmaceutics based research. We do a lot. More or less, you can say we focus on dissolution, or you can say solubility enhancement, where you'll definitely perform a release profile, and maybe you are developing a newer formulation, you would be going for dissolution studies and all. So what I was looking into that, if we incorporate this kind of dissolution data, and if it can, help you out, and we do, of course, what we do is like we perform pharmacokinetic study in animals. So what I thought that, if we go ahead with this, that we use this data set and put it into this modeling simulation, specifically PBPK model. And if we develop that, we can definitely predict. And of course, more or less, we work on the drugs which are generic drugs, and looking for better solubility for them, and kind of, and if we just pick that. So I mean to say that, this all drugs, pharmacokinetics would be already published in human, because the drugs are there pretty much in the market. So what we can do is like, by modifying the formulation, taking out that data, putting that into the software system, and we can predict what could be the pharmacokinetics specifically in the human. And we don't need to go ahead with the animal study and all. And the best part of this work, you just require your laptop, a software, and basic understanding about the science is concerned, and you can do this research in terms of even doing a little bit of the wet lab work or so. So keeping that in mind, it become a boon for us, specifically in the time of COVID, where labs were also closed. So let me just walk you through, before even I go ahead with the case studies. I'll just walk you through. First of all, let's understand what is PBPK modeling. So it is, of course, full form is physiological based pharmacokinetic modeling and simulation. I mean, of course, PBPK. In this, it is basically, you have the computer based software, and in that particular system, you have various like in vivo parameters, specifically your blood flow, or tissue composition, like of the organ. And you can definitely, that how much blood is supplied in that particular, so that is all mathematically put upon in the software. And when you put the data, because we all know the physical chemical property of a drug, that drug is hydrophilic, and you can perform, even if you don't know, you can perform this data in vitro in your lab, which are like pretty much easy way to do what is the solubility. You have the literature data also, but still you perform when you develop a formulation, that what is the solubility in different pH, whether the drug is like partition coefficient, hydrophilicity, lipophilicity. We determine, of course, the log P and PK determination, or that data is also available with you. You can put upon the data like metabolism or permeability related, or how the drug is transporting, if you have that, if you have done in vitro work on that. You can put upon this all kind of a data, that would be as an input factor. And you can say, when you put this all different kind of data set, along with, you can say, various intrinsic factors, like you can have different age, or if you want to check upon that, different race, because we all understand that different race, they would be having different ADME property of a drug. Or if you have a special population like pediatric or geriatric, and you can say like maybe organ dysfunction, and specifically renal impaired or hepatic impaired patient. Or in terms of, you can say, if you want to check upon the effect of alcohol use, or smoking, or some diet on the PK of the drug, then how that is happening. And of course, if you want to know that drug interactions point of view, PBPK model modeling becomes a very useful tool to predict and to make you understand the impact of this all kind of factors, that how that can affect your pharmacokinetic profiling of a drug. So in terms of that, what you have to do, so you can say that you put upon, so this already part of a software, this all data set is there in terms of species wise. In a software you have that data, that whether it is a human or a rat or a rabbit or so, or you can put upon this specifically in vitro data, whatever. So more popularly people put this data of metabolism, drug interactions, or specifically you can say transporter related information like PGP binding is there, or some so this data they usually put upon in the for as a drug property. And of course you have the drug property data when you put in it would, and then when you simulate by putting this info, when you simulate it would give you some kind of plasma drug concentration profile, and that plasma drug concentration profile can predict you or help you, and to know that how this this, and if you change the parameters, that maybe somebody did the in vitro metabolism study, they got whatever values they got, or if someone has done protein binding, whatever information they got, and they're putting in that. So these are like variables, and you can put in that, and you can have the simulated pharmacokinetics, or you can say drug plasma drug concentration profile. And we all understand that when you have a plasma drug concentration profile in hand, you can of course AUC, bioavailability, and that would be very important factor in knowing that how the drug is absorbing, or how the drug is eliminating from the body, and how what is the plasma half life is concerned. And of course based on that you have to decide the dosage regimen, and of course the frequency, it would be coming on based on that. And of course it would be letting you know the if you extend this one, so we call it PKPD modeling, where you even go ahead with knowing that how a drug is actually reaching to the system with site of action, and what kind of effect or efficacy is coming up. So this approach can be, or this is, you can say, roughly called as PBPK modeling. And in terms of I would, because if someone of you have heard for the first time, or if you have little bit heard about it, it is also very important to understand that how this PBPK is actually helping out in drug development, or drug discovery process in today's time. So this is again the data taken from this paper published in JPS in 2019. And of course if you and also in AAPS some of the authors have published, so I have taken from there. And if you just see this are like a kind of like new drug application. So you all are aware that if you are an M.Pharm student, you are aware that after, if you have a drug in hand, if it is a new drug, you would be, of course, new chemical entity, you would be going for all preclinical studies, and then toxicological studies, and then you'll be going for phase 1, phase 2, and phase 3. And post phase 3 clinical trial, you would be going for market authorization application, or we call it as a new drug application. And you would be submitting new drug application to have the market approval or market authorization to launch a drug into the market. Now if you see over here, you can see in terms of the number of counts have been increased, where people have done the submissions of the this NDA application. Over the time, the application with PBPK analysis or PBPK model data have been submitted to FDA, or we can say that Office of Clinical Pharmacology of FDA. And you can see over here a good number is increasing in terms of so they can say, they're saying that almost 50% new drug approvals were actually having the data of PBPK. Now this is a pretty much trend with the US FDA. They are asking this data at the end of phase 1 even, that whatever you have done, so they ask for that. However, not only USFDA but also EMA, which is of course European Medicine Agency, European Union. So they also ask, or they are also accepting the PBPK data based, and giving approval based on that. Similarly, Japan is also in the trend. Recently, Korea has also introduced in their guideline to have this modeling simulation data. India has still not done that, and we really need to think about it. If we are thinking to have India as a innovation hub, then we should also have this kind of approach, and putting upon this data after end of phase one, along with the phase one. If this data is asked by even CDSO, it would be making different dynamics of Indian pharmaceutical sciences is concerned, specifically industry setup or so. So again, these are a little bit more data in terms of that whatever applications were made in this period, what kind of PBPK model or what data set or what they were looking or representing those PBPK model. So either it was for the drug-drug interaction aspect, or maybe for pediatric population aspect, or maybe hepatic impairment. So these are kind of or maybe pharmacogenetic aspect or so, or they had some target population. So this is percentage wise things you can see over here. And of course, in terms of you can see the this numbers have this kind of parameters have changed: hepatic impairment, renal impairment, pediatrics aspect, absorption related, or drug-drug interaction because of the enzyme interactions, or maybe transporter or so. So those kind of approach have been changed in recent time, and the applications are increasing. And this is from USFDA website I have taken. Now, wondering, what kind of even guidance is it available to to follow up on? Because we all know that when you do the regulatory submissions, the guidance plays a huge role to give industry that what kind of a data set is required. And this is USFDA and of course EMA, which are most regulated countries in terms of regulatory sciences is concerned, or regulatory approvals are concerned. So in terms of that, if you see over here, they have particularly in vitro metabolism and transporter mediated drug-drug interaction related guidance document is there. Of course, population pharmacokinetics are also there. I haven't taken over here. How you can go ahead with drug interaction and how you can use that. How you go ahead with PBPK analysis, how you put upon that. And of course, this is from EMA. So this kind of guidance documents are available. This, sorry for this slide, this I have taken from CPT, one of the paper published from by Wegner et al, and and also from one of the paper from JPS. And if you just see this particular slide, it's very interesting slide to see over here. They are saying that regulatory application and predictive performance based on. So if it is a drug-drug interaction, it can be seen as CYP 450, or like as a drug as a substrate, or drug as a, and they have given along with kind of information that what I mean, what kind of publications are there, what kind of a data is available, the whatever research have been done by different people or so. You can see over here majorly drug-drug interaction related, quite some things are there in terms of. And of course we are more interested of course in terms of CYP 450, because we all know that it is a phase 1 metabolizing enzyme which plays a dominant role in metabolism of maximum of the drugs. Now if you just see also in terms of special population and or specific population, so in terms of they are saying that renal and hepatic impairment, so the predictions are there but not that mature, or a lot of information is not there. Of course this is evolving science, so you would see this kind of sentences or phrases. Of course in terms of pregnancy or ethnicity, or you can say geriatric, or of course the pediatric, because still there is a in terms of pediatrics we do have the data related to children, but we do not have a lot of data in terms of infants or neonates are concerned. And I hope you understand the neonates and infants. I'm talking about the population which is lower than 2 years or so. Other than that, what other areas where PBPK can be applied, or you say food or formulation aspect or so. And in this also they are saying that some experience, but it is not that mature data or so. Also in terms of pH effect on fraction absorbed, that how the drug permeability aspect things are there based on the pH or so. And so this is, you can say that, kind of whatever limited information is there, so or good information. So in terms of drug interaction, a lot of people have explored that a lot, and good informations are coming up. And you can say that, to have the labeling and regulatory decisions, because by 2010 it was like majorly it was like that almost 70% of the drug in phase 3 clinical trial were rejected because of the drug interaction. But now that ratio has been reduced quite some manner, because of after 2010 US FDA introduced this white paper related to drug interactions, and transporter mediated, at least for all in vitro data has to be performed or so, and that has changed quite some of the scenario. Unfortunately, in terms of Indian context, we do not have that kind of a data. CDSTO doesn't ask for, and even interestingly, we are more into generic, so we are not even bothered about kind of. But what I'm trying to say over here, that for generic also, when you do this kind of dissolution aspect thing, and if you predict for human, it would be really helping out in a very good manner or so, and it will give you a good data set to understand the science or the drug pharmacokinetics better way or so. So now, just if you see over here, that what data is used, so little bit elaborated in terms of, so you can say like anatomy and physiology, so organism property. So this is species wise, organ volume, surface area, tissue composition, blood flow rates, or expression level of various enzymes or so. Or you can say that when you go for a drug related property, then what kind of physicochemical, because we know that the drug permeation highly dependent on hydrophilicity or lipophilicity of a drug. Of course the bioavailability is highly dependent on you know uh, uh, like what kind of a protein binding is there, or how the drug is permeable. Is it like efflux transporter affected, or it is a substrate for some of the transporter, and the drug is not moving out of the epithelia or so, or it is having some kind of a first pass metabolism, or metabolism factors are playing a huge role also. So you can have this kind of physicochemical property of a drug. Of course, other biological property of a drug are like permeability, or you can say specifically enzyme kinetics if you have that, and fraction absorbed, or permeability in terms of if you have that data also. Other than that, you can say formulation property. So we are working a little bit on here, and as I said, I'm very much baby in this. But yeah, we have explored one drug, and we have communicated that manuscript, and we are learning, and side by side we are presenting also to understand how kind of things are going on and all. So of course, it can give, you have to put upon the data administration protocol, that you have to put upon that what kind of a dosage regimen, dosing regimen you would be giving. And of course, if you have some kind of a special events, like is it a fasted state, or fed state, or exercise, or kind of, so you can have those kind of special event can be even put upon. So you can say in the software, these are divided into three, this kind of a category, that organism properties, or so species related info, or you can say drug properties, or maybe study protocol or formulation properties or so. So this kind of data you can put upon in that, not only limited to that. So this PBPK help you out, you can put upon this all data. And you can have even defining the population, that if you have a data of maybe few subjects, many times it happens that you do not have enough of information, then you have you can elaborate also, or you can say extrapolate it also for population or so, or group of people also. All right. So now coming back to, that was more PBPK, and how regulatory agencies are looking at, and how they are entertaining this science is concerned, and how that is helpful in pharmaceutical product development, or I would say it is expediting the procedure in a better way. So how this dissolution can be utilized, or PBPK model or so. So what we usually do, that we have the in vitro dissolution study you can carry out in different kind of a media, if you just want to understand, that how your drug is behaving or so. Or maybe you can take the same media where your drug is having a good release profile or so, and then you can have this kind of a resolution profile graph, or where you can know the resolution rate and what is the what kinetics of dissolution profile is following for the drug or so. So that can be as an input. And then you can have available literature. So we are talking over here, available literature is of course the physicochemical property of a drug. Okay, we are talking in terms of sorry for my bad handwriting. You have, we are talking over here in vitro, in vivo, in vitro major, like metabolism data. You can put that data, and you can whatever the literature you have in terms of a drug property, as I mentioned earlier, those data you can put upon, and based on that you can have the simulation, and it would give you predicted plasma drug concentration profile. Now what you can do is, with this, whatever the actual data, if you have, in case some pharmacokinetic study have been done, then you can look upon and observe data and predict it. You can have that comparison and you can know. So if you just think about it, you made one different formulation for a generic drug. So if it is a generic drug, of course the pharmacokinetics data is of course available, right? Because it's a generic molecule. So you would be able to having that PK profile and all. And now you made some new formulation by maybe solubility enhancement, or maybe some nanocrystal or something, or a nano suspension. Now what would you do in academic setup in general? That you would do this and you would put this data into, in vivo. You would put this to check upon the pharmacokinetic profiling, and then you would, when you go for the publication, you would be saying that that particle size was reduced, and your release profile was better, and when you had a better release profile, your bioavailability also went good, and you had good Cmax or kind of. So you would come up with that, but you have to do the, of course, animal study in terms of, right? Now what we are saying over here, instead of doing that, you can use that data, put that into the softwares. Of course the software skills are required. It's not like that you would be able to just, it's not just Excel that put the value and you'll get it. You need to understand also, and that takes time. But yeah, nothing is rocket science, so you can definitely learn over a time. And you can have whatever data or prediction or simulation data would come out, you can compare it with actual data, and you can say that whether your drug, what you have developed, or like drug product which you have developed, based on maybe some excipient change, or maybe some method change or reduction in size or so, with better dissolution, you can say that the PK is coming, pharmacokinetic is coming this way, and that would be definitely helping you better way to understand your formulation or so. So in terms of that, I have taken some of the slides from the people who have done some work in this area. I would definitely acknowledge Dr. Jennifer Dressman, who has done a lot work on in this particular aspect, using dissolution data related, specifically PBPK modeling. And I learned many things from her in terms of indirectly, specifically this field. And I could meet her also and had a good discussion about it, because I was doing one project during my postdoc in US where we were doing some kind of this work, not exactly this, but yeah, some kind of this work. So you can see some of the examples over here, and this is recently, like just 2020 the work was published. And if you see over here, you can see that BCS1 or class 3, BCS2 or class 4, class 2 4, you can see various drugs are there, losartan, and all different kind of drugs or compounds are there, and dissolution modeling approach. So they have taken the dissolution data and they have put upon in the information, and develop this PBPK model and check upon how things work. So you can see this reference later. So from that only, this is not, I think it was some different paper, yeah, from Journal of Pharmacy and Pharmacology. It's one of the old papers where PBPK was very not utilized that much. But this is very interesting example. If you see over here, this particular drug, you can say they did the development of some nanocrystal. Okay, they have jet milled product, and they have used the hammer mill. So we are aware of that, when you do this, basically you are reducing the particle size, and we understand that when you reduce the particle size, of course over here the particle size would be higher. These are the nanocrystal, particle size would be lower when you have a particle size. So you can see over here, so this is a fasted state, this is a phase state. If you see just over here, what it reads is basically, the solid line represent the prediction from the particle size model for a mean this psilocybin plasma clearance, and the gray shade area indicates the variability of the plasma concentration due to inter-individual clearance variability. Now, okay, if you are reading this kind of thing first time, so I just simply tell you that what is exactly about. If you see over here in terms of this is particularly nanocrystal, and this is jet milled product. Okay, if you see over here, you can see that there is a decrease in a rate of absorption. So this is the rate of absorption, right? Decrease in a rate of absorption with increase in particle size. So this has been predicted by the model. You can see over here, this is a model prediction that, of course, and this is of course the fasted state they are talking. Now in phase state, drug has better better absorption, but of course in terms of, you can say, what they are saying, that in phase state, again it looks very kind of a similar sort, not similar, but again it has specifically low plasma concentration in jet milled and hammer milled than nanocrystal, and you can see the impact of fasted and phase state. So this is just based on the dissolution, and I hope you all of you are aware of or have heard of biorelevant media. uh, when we Sorry about this. When we talk about biorelevant media, we are talking about not only just pepsin and trypsin things are added, which is generally your simulated media, what you call it, but biorelevant media where you have actually bile salt also, like sodium taurocholate and lecithin have been added in that, which have been developed by Dr. Jennifer Dressman and her team. And pretty much it is very much utilized by a number of companies across world, where they are simulating. Because what we do generally, we do the dissolution studies in the pharmacopoeial buffers, or you can say maybe acidic or basic buffer, that generally we follow, or trend. So now what we say, that what we do is actually we are checking the formulation property, that whether it has a quality control parameters are in compliance or not. A drug is releasing within certain period of time. If it is within like linear release is there or so, we just check upon that. Or what kind of a order of kinetics it is following in, or what is the dissolution rate or mean dissolution time of a drug, we can know that. Now, in specifically in terms of, if you go for biorelevant media, which is actually exactly simulated media, what they have developed, and in that if you do, it would be giving you a better idea that there won't be a drastic difference between your formulation, like dissolution related, and in vivo performance of a drug product. You can have biorelevant, at least it would be pretty much coming closer to your in vivo prediction or so. So we were working on actually this concept in terms of, so this is another case study, and I'll walk you through that, the second one, what I'm talking, bile salt related, in a while, or biorelevant media related. So basically, if you see over here, this is the second example from again EJPB, this Jamai et al. And what they have done over here, so they have a reference tablet formulation. So this is reference, formulation tablet, normal conventional formulation, and then they have this chewable formulation. So you can see that the kind of percent dissolved is is higher in chewable formulation, because the release would be very rate controlled in terms of or so. And if you see, they have this again, they are just checking the precipitation inhibiting effect of this methyl cellulose, which is one of the excipient they're looking into, and which is impacting actually this dissolution of the drug. And if you see over here, they have without excipient how the drug is having, and if you see in terms of with excipient, how the precipitation behavior of this particular drug, because this low viscosity methyl cellulose what they have used. Now if you put this data into, authors have put that data into, this model, and what they found is that, look upon the plasma drug concentration profile, or rate of absorption. So initial first 30 minutes itself, you can see the substantial difference, and this is actually a predicted, you know from the formulation. So actual PK is not done. This is actually predicted one, and it can give you this in vivo results, that and this is of course the variabilities are there in terms of. So this kind of, and they have used a biorelevant dissolution, what I mentioned, biorelevant media they have used. So dissolution rate is of course different, and how that dissolution rate difference is there, you can predict it through the software. Even without performing in human, you can do this and understand your product better. Now you feel like, okay, that maybe you want to play a little bit more, you can change your excipients, and you can work out, and do the PBPK modeling, and understand or make the product better in terms of a release is concerned. So this is a second case study. The third one is actually done in our lab, and specifically, we have utilized one of the software over here. So one of my student, PhD scholar, she is her name is Miss Priya Sharma, and she put a lot of efforts in bringing this on slides, I would say. So specifically, the drug voriconazole. So I was working on this drug when I was doing my postdoc, and this drug is very interesting molecule. In this particular drug, this is for systemic fungal infection. This drug has almost 90% bioavailability in adults, and in pediatric population. So pediatric, we mean 2 to 11-year-old children. The bioavailability is 45 to 65%. Now what we do is, usually for children dosage regimen, we just reduce the size, dose calculation by means of Young's formula, or body weight and surface area and all, and we do that, and considering miniature adult children as. So our lab, my boss, former boss, Dr. Devin Thakur, was, he was, we just share a common survey. So basically, his lab was looking into this, and when I joined, so my role was, to understand, because the drug is lipophilic. So what we're trying to look into, that if the drug is lipophilic, then of course the bile salt proportion in pediatrics, if it is less, or in adult if it is high, then of course in high bile salt, drug is lipophilic. So bile salt would help as bioenhancer and help drug to permeate more, and you would be having better bioavailability in adult, while in children it might have not, because children have usually low bile salt concentration in their GI or so that was the concept we came up with. And then I was working, I did some ex vivo, so we took out some of the mice, young mice, and adult mice, we considered that way, and we did some permeation experiments doing, and we could come out with that. So what I thought at that time, it was actually permeation data we were doing. So over here we did actually the dissolution of this. We took out the different bile salt containing media. So if you have a biorelevant media, for adult, you have the concentration of sodium taurocolate, what I mentioned, has 3 millimolar. However, in children it is 1.5 millimolar. Okay, so we have this two different biorelevant media. We took that. Um, I would say we did dissolution data in quality control buffer, QC buffer for a drug, and of course the biorelevant media. We had this different bile salt concentration, and of course, considering pediatric, they have a low volume in the stomach. Considering we took GI volume is 500 ml, and of course, when volume difference is there, the release profile or pattern will be different in terms of, or dissolution rate might be different. So we put that information, and we incorporate this dissolution data along with. So if I go to the previous slide, along with, you can see this dissolution data. This was the parameters we took from the literature. And of course, there was one paper which was published by Zain et al. And we took that data specifically, you can say log PBPK and all those data. We put this information from the literature, and we did in vitro dissolution. And actually, this in Dhiren Thaker's lab, they were doing in vitro metabolism data also. So we took that data, we put all those information, and you can say what was done, that all this information related to first adult model we developed. So develop and validate the baseline adult model by comparing against with the published value, and we validate this model, whatever the published literature, and then extrapolate this model, adult model to pediatric model. That can be done by the software. And then for pediatric, we put the pediatric info, what we had from the literature and what we had in our lab, like what we did in our lab is diso-related data also. So now what happened when you do the simulation? So in terms of, if you just see over here, so we comparison of simulated pediatric plasma drug concentration profile specifically, and we found out that when we developed from the base model to pediatric model, we had prediction error was less. I mean, we could validate the model based on the adult model. And if I go ahead with a little bit in terms of that what kind of PK analysis for pediatric it has happened, because we not only just comparing if you see over here, this three different table you can see over here. So one is without disso. So in not adding dissolution data, just adding the you can say the data of physicochemical property of a drug, that log P or literature value, no dissolution data. So if you see over here, Cmax was coming 4.74. Now when we add dissolution data over here, you can see we have with QC buffer it is coming 2.36 Cmax. Now however, when we go ahead with biorelevant, again it is, now look upon the difference. If you do not use biorelevant media and if you use biorelevant media, there is a huge difference in your Cmax is coming up also. So and this is again, you can see over here that this is QC buffer and this is biorelevant media. Look upon the quite significant difference in the release rate. This is actually a dissolution data, and you can see significant differences there when you have the release of a drug in QC buffer, or you have it in sorry about this, and you have it in biorelevant media. So what we are trying to say that it's really not wiser to use quality control, significant decrease in predicted Cmax in biorelevant media comparison to predicted Cmax. So, it says that, there is, this kind of trend is we are observing, and it won't be wiser to use quality control buffer rather, with the dissolution data, you should prefer biorelevant media also. This is further, adult and pediatric model comparison. So we did that. If you just see over here the difference in the Cmax, you can see how much it is coming up also. And, so this difference you can see, it's significant decrease in Cmax and AUC also. So basically with this work, we could say that, QC buffer is not a it can give you a misinterpretation of the results. Rather, you should use biorelevant media, and more or less many discovery companies does that. It's not new, they use it. But what we were saying, that using even that data, if you incorporate into the PBPK or develop that model, it would give you advanced or better information about your formulation also. So in a nutshell, we can say dissolution data in PBPK modeling, it can help, formulation development. Early stage, you can know about it. It can give prior information on how food or excipient can affect, how much particle size or dissolution on PK profile, how specific, it allows specification changes, prior approval or post approval, and it can give you a good database for bioequivalence waiver or so. So this can be helpful. Various softwares available, Pumas, because I always put this software first. I know it has been, it has been invented just few years back. There are player before that, but as I started learning things from Pumas, so I always put this software first. So you can say non-mam, Monolix, Phoenix, you have PK Sim. We have done all the work using PK Sim because it's free available. We are going to try with Pumas also. We are in talk with Pumas persons, specifically Dr. Vijay Vaturi, who is from, who is co-founder of Pumas. So probably we would be able to use it. I did little bit work on Gastro Plus, Simulation Plus, SimSip. These are again, I would say in India, specifically CBRI Lucknow, you would be able to see they are using it, or in terms of your Setara is also there, they use it also. Am I audible?
Yes, ma'am.
Okay, all right. MATLAB is there, WinOnline is there. So these are the various software. Oh, okay, so do we have time for 5 minutes? I can just talk a little bit on PKSim. Do we have time? 5 minutes?
Yes.
Okay. I'll very quickly switch to this, how you can use this PKSim. Um, we are going to organize some workshop on utilizing, PKSim. So if anybody is interested, just keep a track of my LinkedIn, so you would be able to see it. And specifically, when you use PKSim, it's again software by one of the known company, Biotechnology Services, Germany, and they have developed this software. Interestingly, this software is free available. You can use it, but yeah, of course, you need to learn how to use it. So you can see over here, you would be able to see this kind of. So you can add the data of individual. What you have, you can create a population. Like if you have in one group, like if you say you have rats, if you have done studying rats or rabbits, you can put like in one group, if you have 4 to 6 maybe animals, or maybe 6 to 8 animals, you can put that as one group and population. You can put the data of compound, so you can see this, now what data you can, so this is over here, it says human, but you can change the species, and you can put this all information, that all, so more or less all this information would be available. But in case if you want to add something, you can do it. And these are specifically, you can say, expression of enzyme. If you are, some reference concentration, if you want to put upon the data, what is, like somebody else have done some pharmacokinetics of that particular product, which you have also developed as a new dosage form, you can put that as a reference value over here. And or maybe some metabolism data, if you can put those value over here, and you can give the reference and all actual values and all. Uh, for the compound, so you can say, so similarly you can do, as I said, that same information you can put for your population. If you go ahead with the compound, in that you have all information, measured, or literature. If it is literature, so value, you can put this, that is it manual or from publication, so you can put the publication actually over here, reference over here. And in terms of what kind of a binding is there, And again, from where this value has been taken, you can put that. Molecular weight is there, or all those information can be put upon. You can see that is basic physicochemical property. Then you can go ahead with ADME, and then you can have the advanced parameters. So if you see in terms of various metabolizing enzyme related data, you can put that. Of course, in terms of now you come as a formulation. So what your formulation is actually, so you can put that data about your formulation. Along with that, when we talk about dissolution, that in dissolution we have various empiric function, or what kind of dissolution rate, and different methods or models are there. Linked 80 means it is like 80% of the drug have been released linearly, so you can put that. Are you looking for particle related dissolution? Then you can put that. Is it following zero order or a first order dissolution kinetics? Then you can put that. Or you have Webull. Generally we prefer Webull because it is very largely utilized method since almost 1951 or so. So we usually take that, and you can name the drug and all information about that. And of course, administration protocol, that how this dosage was given actually. So what dosage regimen, if it is animal you can put upon, or any human, if you are putting that data you can put that also, or literature data also. And then you can have you can create and compare this simulation, because you have put the data of other published thing also, so you can have that comparison, and you can have this kind of a plasma drug concentration profile. And you have this data set also. Of course, we are going to have a elaborate maybe a workshop, because this is a free software, but a little bit knowledge in this, you can change your publication system, and you can have this can be done also. Anyhow, with that, I'll just wind up. This is my PhD. These are my PhD scholars. Interestingly, they are working in different aspects, because my specialization is QA, so I'm dealing with regulatory sciences also. More or less, all are part-time PhDs. We have corporate PhDs also. So uh, uh, you know, he is Mr. Rajendra Singh. Sorry about I haven't put his name. He is vice president in Mankind. We have Miss Nisha Kaushik, because I have people from CDSU who are also pursuing PhD with me. Now Mr. Ranga is deputy drug controller in CDSU, and Miss Anjali, she is again faculty in Amity University Haryana campus. She is also working on formulation development, and Priya, she's working on this modeling simulation related project or so. And last slide, thank you. And I would say I'm very inspired by late APJ Abdul Kalam sir, who says that dream is not the thing you see in sleep, but is that thing that doesn't let you sleep. And my two little bundle of joys, who always put me on toes, that I'm not just only a researcher or a academician, but I'm a mother of two, so I have to be balancing out that. So with that, thank you so very much, and I'd be happy to take any questions.
Thank you so much, ma'am. It was a very informative session. If any participant has any questions regarding this, can ask or write down in the chat box. Any questions?
Good afternoon, ma'am. I'm Dr. Vivek Srivastava. Am I audible?
Yes, you are audible. Yes,
Yeah.
sir, you are audible.
Thank you very much for very comprehensive talk on this topic. This pharmacological based pharmacokinetic modeling. Madam, how relevant these models are for ANDA submission also?
Of course,
You
they are
were talking
relevant.
about NDA, and there is a
Yeah,
repeat surge you told. What about
yeah.
ANDA, madam, in that case?
Of course, for ANDA also they are doing, in terms of when we go for IVIVC, even they are doing some of the data is entertained, but that is even in terms of ANDA. I would say more would be population specific, pediatric aspect or so. So those kind of still submissions or approvals have been given.
Now, once you validate, once you develop, do you need to validate the model?
Yes, so this validation of the model is done usually based on the published pharmacokinetic data. So what we do usually, we initially develop a method model based on published data. So or you can say the literature data. We will call that as a base model, and then we will put our experimental data in that, and then whatever the prediction would be. So you have the base model. With base model, your experimental data related model, you can have the comparison, and you can validate based on that. And that validation is done usually by means of percentage error. So that values are predefined, that this percentage error is less than 30% or something, then your method is considered to be validated, or model is considered to be validated.
And then also there is some racial differences so far as pharmacokinetics is concerned. Like the Asian population or the what you say the other population, there may be some variation. So this
Very good
software
question.
is considered.
Yes, yeah. So very good question. I don't think all software had that kind of info, but I mean, we were even as I was saying that one of our collaborator, we had a discussion with. They are actually looking into. There is also one more person working in Tata Research Center or so. But this is really a good question, and kind of a point where we need to understand that how Asian, or specifically Indian, because we are like what, one third of a world population, and whatever the database are there is in software is. But in this, the prima facie, you can put your Asian, like Indian clinical data. If you put that, and that can be compared also. So yeah.
And my last question is that on the that is from the drug that you were talking about, voriconazole, and there was a
Uh-huh.
drastic difference between the adult population and the pediatric population. And
Of course.
you, what I should say, you resolved that matter by predicting that bile salt concentration.
No, so
There is a
it is
difference
not
in bile salt.
purely. Yeah, so it is one of the reason that bile salt are different in pediatric and adult. So that is also one of the factor, but that's only not the factor, because I didn't talk about it. I did other project where I was working on that metabolizing enzyme, because in pediatric, the expression of for the enzymes which are there in pediatric are different, compared to. So you can say ontogeny of enzymes are different in pediatric and adults. So voriconazole has impact of this metabolizing enzymes also in pediatric and adult. So FMO enzyme is not involved when you go for adult metabolism of the drug. It is not involved. However, in pediatric, it is involved. So we can say that over there, extensive metabolism of a drug is there, because there is one more enzyme is involved, and drug is getting metabolized more, and that's why it has a low bioavailability, and a non-linear clearance is there.
My question is there any transporter specific for this particular drug? Because there may be
No, there
difference
is
in
no
either
transporter. It
receptor
is no
expression.
transporter. No, no, sir. I think if you understood what I just talked, it's metabolism aspect things are different. So if I say in pediatric, there are different enzymes are getting doing metabolism of a drug. So just think of, if you have more uh, enzyme or different metabolizing enzyme or extra metabolizing enzyme in one population, and in adult that enzyme is not doing metabolism of a drug. We did that study actually in my lab. People were working on that, and that's what the drug extensive metabolism difference was coming up. There is no role of transporter yet we have seen, or there is no any kind of info in that. Drug is a lipophilic molecule. There is no any other transporter involvement at all. Drug is having simple permeation, but drug has a extensive metabolism, first pass metabolism. So if you say in adult, expression of enzymes are there, compared to that in children, extensive metabolism is happening because more enzymes are involved also. So that's, I
My,
would say, both reasons are there: ontogeny of
my
enzymes and I think, sir,
Why
still
I
you
was
didn't
asking this
understand because I.
question? No, I
Yeah,
understand,
yeah.
but why I was
Say that.
asking this thing was because, madam, in the second wave of corona, there was one
Hmm.
brief that coronavirus was hitting more in adult population as compared to the children. And the
Hmm.
reasons they gave for that particular thing is the receptor expression. That coronavirus, the spike proteins they bind to the receptor, so those receptors are more expressed by adult population as compared to the pediatric
Sir,
population.
these are
That's
two
why corona
different
is.
things. No, these are no, no. These are two different things we are talking. You are talking related to, if I talk about voriconazole, I'm talking in terms of efficacy wise. There is no issue of because these are two different things. When we talk about pharmacodynamics, efficacy, this is different story. I'm talking about bioavailability of a drug. In adult it is higher, in children it is lower. So we are talking expression of metabolizing enzyme is different in these two population, and that's why you are having difference in the bioavailability. However, what you are saying for corona, in terms of expression of receptor or site of action point of view, voriconazole doesn't have any pharmacodynamic related issue. It is functioning very well with both population. Only difference is coming up, bioavailability is different, and that bioavailability is different because of the two reason. One is different, pediatric have different bile salt, so ratio of that is affecting the permeation of a drug. Second thing is, whatever the drug is permeating, it is extensively metabolizing in pediatric compared to adult, because expression of enzymes are different in pediatric compared to adults. So I hope that.
Okay.
Yeah,
And madam, what
yeah.
is the cost of Pumas?
It's, I think, free for some 400
Pumas is also
hours
free.
or so.
Okay,
Yeah,
okay.
it is
PKSM is
for
free and
academic. Pumas is also free, but Pumas has
okay.
some 400 hours. It is free, and we know academician doesn't need more than that even. That's quite some of thing. So yes,
Okay, okay. Thank you.
thank you. Anybody else? Any question?
Anyone? Thank you, ma'am.
Thank you.
As a token of appreciation, I would like to felicitate ma'am with the e-certificate on behalf of our university that is accepted. With your
Thank you.
permission, I would like to share my screen, ma'am. Scan.
Um,
Thank you, ma'am.
all right. Thank you so very much for having me here, and stay safe, everyone. A wonderful time.
Thank you, ma'am.
Thank you.
Have a nice day.
You too. Thanks. Bye bye.
All the participants will receive feedback form link and all of you need to fill it.
All right. Should I leave?
Link. Yes, ma'am.
Thank you.
Thank you. अब क्या करूँ?